Hello, this is Mr. Arthur. Welcome to our lesson on Higher Unit 1, KBR2, and we're looking at PCR. So this is a PCR machine. It's used to amplify specific target sequences of a DNA molecule. Much like mitosis, that means it doubles the number of molecules every cycle that the machine does. And now, a common misconception might be that things are being added at the different stages of the PCR reaction. But in fact, everything's put into the machine at the start. So the DNA to be copied, the free nucleotides, the primers, the enzyme, they're all in there at the start. Okay, so these are the list of things that we need to know about PCR. The reason why PCR is used, the 3 different applications of PCR. We need to know the different temperatures at each stage of the cycle and what's happening at these temperatures. And you need to be able to do this maths like mitosis to be able to work out how long it would take to amplify a specific number of DNA molecules, or calculate the number of molecules after a given number of cycles or a given time. Looking at the course notes then, we're going to look at the role of primers again. We're going to look at this term to amplify. So PCR is repeated cycles of heating and cooling, and its purpose is to amplify the target region of the DNA. You can see here that there's a range of temperatures that we can talk about. So the first one is between 92 and 98, and that's to separate the DNA. Now it's key to PCR that it's 2 single strands of DNA being replicated. It's not a replication fork like DNA replication inside the cell. So in your head, be clear the differences between the human scientific process in the machine using single strands to amplify a target sequence, and how that's different from the leading and lagging strand which goes on within actual living cells before cell division. Okay, so repeated cycles of heating and cooling. We've covered the first stage. Second stage is it cools right down to between 50 and 65. That allows the primers to bind to the target sequence within the DNA molecule. And then we heat it back up to 70 to 80. And that's because that's the optimum temperature for enzyme to replicate. Enzyme's got a similar name. It's DNA polymerase, but we're going to call it heat-tolerant DNA polymerase. So it's important to use the full name when it's a PCR question. Okay, so what's the point of amplifying all this DNA? Well, we can use it to solve crimes for DNA profiling. We can use it to settle paternity suits, and diagnose genetic disorders. Okay, so a PCR chain reaction, first of all, in red, the new key term, it amplifies a specific target sequence of DNA. So here we have 2 DNA sections. I've highlighted the target section in red, so this is the part of the DNA molecule I want to amplify, and I've designed 2 primers in green, which will bind to the 3 ends, which allows the heat-tolerant DNA polymerase to replicate and copy these target sequences. Now note the 2 primers are different. That's because the 3 end of the top strand is different from the 3 end of the bottom strand. So in PCR, we're talking about 2 separate strands. Both of them have a different 3 end, so both of them have their own primer, and that allows both of them to be replicated. There's the cycles that we covered already. So 3 steps really. Up to the high temperature, that breaks the hydrogen bonds between the 2 DNA strands, and it separates them into 2 separate strands. So that full sentence is required. Breaks the hydrogen bonds, separates into 2 separate strands. Cool down to 1565, that allows the primers to bind to the 3 ends of the target sequences on each of the separate strands. And then between 70 and 80, that's the optimum temperature for the enzyme heat-tolerant DNA polymerase to replicate from the primer onwards. Okay, and that's what we can see in the machine here. In the little PCR tube, we have to put in the TAC polymerase, that's the heat-tolerant DNA polymerase. You have the nucleotides with the different bases, they're free in the solution so that the heat-tolerant DNA polymerase can add them to make a new strand. It requires the 2 primers before it can amplify the section, and we need the original DNA sample that has our target strand in it. This is all mixed together in a liquid called the buffer. Okay, so there's a key word we can use at each stage. Stage 1 is denaturing. You can describe the DNA as being denatured. That saves you writing the whole sentence to break the hydrogen bonds and separate into 2 second strands. Annealing is just a fancy word for the primers binding to the 3 ends. Extension is the heat-tolerant polymerase replicating the target sequence. That happens 1 cycle, and it happens again, and it happens again, and it happens again. and we can see that here. We start off with one molecule, two separate strands, primers bind to both three ends. Heat-tolerant polymers can then extend and replicate and synthesize a new strand of DNA, so we have two new molecules at the end, and these two new molecules will then both get replicated in the second cycle to become 4, third cycle become 8, fourth cycle becomes 16, and so on. Here's another way of looking at it. The first cycle on the left, we start with one DNA molecule, at the end we've got 2. The second cycle we start with 2, at the end we've got 4. So this is showing the doubling and the doubling each time. It's also showing the same process. Denature the DNA, allow primers to bind to the three ends, allow heat-tolerant DNA polymerase to replicate a new DNA strand. Okay, so this is our first slide again. Our primers are still there. Because the primers are in place, if we increase the temperature back up to about 75, the heat-tolerant DNA polymerase can work left to right, replicating that orange strand. Okay, and same in the bottom, it's working right to left from the 3 to the 5, and it's replicated it. So we've ended up with 2 copies of original DNA target sequence. It's important to use the word target sequence in PCR because the primers are designed to bind to the target sequence. It's not the full molecule that's replicated. Okay, it's easier just to remember a temperature rather than trying to remember the range. And I try to remember them all just with 5s to make it easier. So 95, so that's almost 100. So I think 95, about half of this, but still sticking with 5s, is 55. And then back up to halfway between 55 and 95, which is 75. So easier to remember because one number for each, and they all end with 5s. 95, 55, 75. 95, the DNA denatures. 55, the primers bind to the three ends of the target sequence. Back up to 75, as that's the optimum for the heat-tolerant DNA polymerase to replicate the target sequence. After that, it's going to go back up to the top, and when you see it go back up to the top, that's the end of one cycle, and it's going to start again. So whenever the pattern repeats in a past paper question, that tells you that it's on to the second cycle. And this is what we're seeing here. On this diagram, there's actually 3 cycles of PCR. You can see that there's 3 peaks and 3 troughs. So the first thing we need to do is work out how long one cycle is. So the cycle starts straight, then it goes up, then down, then back up, and then goes straight for 2. The second of those is the start of the next cycle. So therefore, I've put the blue line there to show that it's 4 minutes. Double check this by going to the start of the peak. The start of the peak's at 1. The start of the next peak is at 5 minutes, so therefore it's 4 minutes between the start of the peaks. So one cycle is 4 minutes. The question says if there were 500 at the start, so write down 500. Predict how many there'll be after 20. So we know these 500 are going to double to become 1,000, 1,000 doubles to become 2,000, 2,000 doubles to become 4,000, 8,000, 16,000, 32,000, 64,000, and so on and so on and so on. So we know this is what's going to happen, so we can start to write down the DNA, show that it's doubling each time, but we don't know how long to go for. So the question says it's going to take 20 minutes, and the graph shows us it's 4 minutes for one cycle. So in 20 minutes, you should be able to work out you're going to get 1, 2, 3, 4, 5 cycles. So therefore, always remember the 500 is before the PCR, so that's 0 minutes. 1,000 is 4 minutes, 2,000 is after 8 minutes, 4,000 is after 12 minutes, 8,000 after 16 minutes, and therefore 16,000 after 20 minutes. So that's our answer. Okay, practical applications. If you ask for the application, it's different from the purpose. So the purpose is to amplify DNA. The applications are to settle paternity disputes, that's who's the father, to diagnose genetic disorders, and to help solve crimes through DNA profiling. So here we've taken a sample of DNA and we've run it through an electrophoresis gel. And what that does is it separates out the DNA into these bands. Now we can compare people by looking to see which bands they have. If you're related to someone, you're going to have the same bands as them. You get the same with a crime scene. So we've got the victim and we've got the crime scene blood. Now if you look at the crime scene, the first band is the same as the victim, so that may be the victim's DNA, but the victim doesn't have that second band, so that suggests it's from the criminal. Third band matches with the victim, but then there's a further band at the bottom the victim doesn't have. So we're really interested in the second and fourth band on the crime scene. Looking across at the suspects, only suspect 2 has that bottom band. Okay, so suspect 2 must be the person who's also contributed DNA to the crime scene after we've removed the victims. Again, the child must get half their DNA from their mother. So looking at the child's bands, let's see which ones come from the mother. So the second one comes from the mother, so we're going to put a line through that and ignore that. The third one comes to the mother, we can ignore that, and the bottom one comes to the mother, so we can ignore that. So we're really interested in the first band and the fourth and fifth bands. Now if we look across, Dad 1 only has one of those bands. Dad 2 has the first band and has one of the 2 bottom bands. Dad 3 has the first band and both of the bottom bands. Therefore, Dad 3 is most likely to be the father. And here's a past paper question asking that. So in yellow, when you're reading a past paper question, it says paternity suite. That's KU to you. That's an application of PCR. That's finding out who's the father. Again, look at the mother and rule out the mother's bands, which I've done in red, and that leaves us the 2 in blue. If you're asked to write a conclusion, there's several things you could say here, okay? So you're trying to find out who the father is. So first of all, I would say that the father is going to be D because the father has 2 bands which match the child, whereas father B and C only have 1 band and father A has none of the bands the child has which have not came from the mother. Although we're looking at DNA, sometimes this can throw up an odd question. It could look at the protein because the DNA codes for the proteins. So if you get the same thing, if you break a protein down into amino acids, you can look to see the bands that match. Down the left is a protein standard. These are standard 6 proteins that we know. We can see that all 4 species have protein 2. And what we can see is the more they have the same, the more related they'll be. So species 1 and 2 share 3 proteins that are the same. Species 1 and 4, 1, 2 and 4 all share 3. So they're more closely related than species 3 because those species 1, 2 and 3 must have the same gene coding for the same protein. So they're more related. Okay, here's some common questions then of what we've just covered. The purpose of PCR is to amplify. Enzyme involved is heat tolerant DNA polymerase. If you're asked why we're using heat tolerant, it's because it won't denature during these repeated cycles of heating and cooling during PCR. Applications, easy one to remember is the paternity test to identify the father. Because you're looking at DNA sequences, we could use it to identify genetic disorders. I would use these first 2 and save DNA profile and solve crimes as the third option because it's a bit vague. Explain what happens at each temperature. So, remove the 5s, 95, DNA molecules denatured. 50, primers bind to the 3 end of the target sequence. Back up to 75, it's the heat tolerant DNA polymerase replicates the target sequence. And that's the optimum for this enzyme to work at. You can pause the video here and read through the checklist of all 3 parts of this unit from the SQA. And then come back to some past paper questions.